So I'm going to talk to you about quantum mechanics. And quantum mechanics and biology normally are not two, they're both forms of science. They're not normally mentioned in the same breath. So we have the beautiful background here of a biological environment in Yellowstone. Now, where is the quantum mechanics there, you might think? Well, so there's actually a lot of current interest in exploring possible instances of true quantum behavior in biological systems, ranging from plants and bacteria, photosynthesis, which I'll spend most of the time talking about. This sort of drifts. To birds. I'll tell you a little bit about birds. This is kind of like an ant. To maybe I can do it with this. Nope. Oh, here we go. Yes. Is it there? Right. Okay. Birds. How birds navigate north and south around the globe. To smell. Animal senses smell. I won't have time to say anything much about this, but if you ask me afterwards, I can say a few words. And ion channels, and in the brain, there's been lots of discussion over many years about that. So the resurgence in this really is a resurgence of an interest that started a very long time ago. In fact, Bohr, when he'd cleaned up quantum physics, turned his attention to biology, and he thought that quantum mechanics should apply to biology, and spent basically from 1929 onwards thinking about this. But the first person who really actually made a serious contribution here was a student of Bohr's, Max Delbruck, who was really the first quantum biologist, and he was successful because he worked together with biologists. Always useful to actually have an interdisciplinary team to deal with an interdisciplinary subject. So he got together with a geneticist and a photobiologist, which is now what nowadays you would call a physical chemist. And they probed what at that time was called genetic structure and the mutations and stability of that genetic structure with X-rays. And this was the first true quantum probe of biological structures and function. So this basically put on center stage the need to understand the detailed molecular structure of functional biological systems. And so I tend to classify the whole field into an AL and a BL. The BL, like BC, is the first era, and the BL means before the laser. And this is the era in which people recognize that biological systems are made up of atoms and molecules. Atoms and molecules, we understood from the beginning of the 20th century, were quantum mechanical in nature. There were quantized energy levels and energy barriers, and the stability of biological matter could be understood in terms of sort of the basic quantum elements of matter. And that's all nicely summarized in Schrödinger's book in 1943. But the second era, so if we like, the culmination of that would come in the 50s with the structure of DNA, the molecular structure of DNA. The second era, however, starting in the 1960s with the laser, so I call that after the laser, is an era where we're focusing not on structure and stability, but we're focused on quantum dynamical effects, and where this interest is being fueled by new generations of dynamical probes coming from nanoscience and nanotechnology, and a lot of innovation via the new quantum science and technology, which is linked to things like quantum information, quantum computing, and so on. So this is a summary of the kind of things that are going on today that I would classify as developing new probes of structure and dynamics of biological systems. On the top is just one example, where a group at Harvard has a surface which they put these tiny little silicon nanorods on top, and then they just deposit living cells on top of this, and they use the nanorods as a tool to access the cells, and they can deliver biochemical or biomolecular probes into the molecule. They can look at the cellular response with electrical monitoring and so on. The other form is a form which lies behind the kind of work that I'm going to tell you about in photosynthesis, is are very sophisticated forms of spectroscopy where we shine light on samples, and in this case we might shine 3 different rays of light on something all very carefully timed, all with specific spatial directions and phase matching and so on. And that enables us to learn something about the dynamics of electronic energy in photosynthesis. So if we were looking at the big picture, why are we studying this kind of thing? Well, we could say that we're driven by the tools, which are the tools of quantum science and technology, which enable us to come down and down and down in the length scale. So going from the whole brain, for instance, down to single neurons, and then further down through the ion channels and individual molecules and atoms. So down with these tools, we can go to areas where we know, when we're down to individual atoms and molecules, that quantum effects should be important. The real challenge is going up again on the other side, which is the side where we schematically here represented function. So we know that there's somewhere here on this two-dimensional plot, time scale and length scale, there is a region where everything is quantum. Coming out here, normally we can just take the classical description. But what does this tell? If we come out here from classical up to larger and larger length scales and larger time scales, what do we learn about having probed this quantum region here? What do we learn that's essential to the biological function? And this is, I think, a true unsolved problem for any given instance of quantum biology that I can show to you. It's an open and very challenging and very exciting question, namely whether the quantum behavior of the molecular and atomic dynamics can be relevant for biological function. So, and this is not an isolated problem in biology, it's very much more unusual in biology than in physics. There's often a discussion about the quantum classical border. When do things behave quantumly? When do they behave classically? Typically, we think about the quantum world as being the microscopic world, electrons and atoms, and classical behavior would be the behavior of a pulp like this, macroscopic object. And in physics, we have the same sort of incongruous situations where often people are looking now at macroscopic objects, like this world here. We go to some place here, and is there a sharp boundary between a classical landscape and a quantum landscape, which is just schematically indicated here by these paths bifurcating the clouds, dividing into two, and if you know about Schrodinger's cat, it is having a nap, and an eternal nap down here at the same time, so on. All right, so that's just a cartoon. But that's to tell you that this is why this quantum biology is of great interest to physicists as well, because they are definitely what we call open quantum systems. They're not isolated biological systems. So. Right, so I'm going to say a few, probably most of the time I'll spend actually summarizing the effects that have been observed and our analysis of this in photosynthesis. Then I'll say a little bit about the birds, and I think that'll probably be all I'll have time for. So in photosynthesis, so on the right there is a little cartoon of what photosynthesis does. A green leaf would absorb light from the sun, and this light is converted into electronic energy by these objects called light harvesting complexes, and that's these shown here for purple bacterium, these beautiful little circular structures. And the electronic energy is then used to drive chemical reactions which build up the chemical storage, or the chemical storage of energy, which we then use, or the plants use to survive. So this light harvesting is an amazing, it's actually a truly amazing process. The energy travels over about 30 nanometers in 1 nanosecond. It's very fast. And I've lost my, yeah, here it is. And it has near perfect efficiency, meaning that for every single photon or every single packet of light that comes in, one electron essentially is created and can undergo a chemical reaction. So there's a quantization condition here, and it's an efficiency which is very, very unusual in a biological system. I mean, these things really have 95, 98% efficiency of conversion of a photon into an electron. It's actually an electron hole pair. Secondly, the experiments reveal a kind of wave-like transport of the energy. And it's this wave-like transport which has got a lot of attention in recent years. And you can think of it just in a non-scientific way of saying that the excitation energy is spread out over a rough energy landscape. And if you have a wave, it's like having a big football which goes over a rough landscape much easier than a golf ball, which will get stuck in these traps between these sites here. And these are the things that we found out about this. This coherence does contribute about 10% to efficiency. The natural systems are amazingly optimal as far as we can see with respect to all possible parameters in the system. There's no artificial system that gets anywhere close to competing with this high efficiency of basically just harvesting the photons. Another interesting thing which we don't fully understand why it's there or whether to what extent it's general is that this coherence does enable you to go uphill, so that's actually related to the roughness of these surfaces here, and also does enable long-range energy transport. So, so let's see, so a few more interests. So this is again just a summary. So here are the light reactions. So this part here where the quantum behavior has been evidenced is just up here where the light energy is absorbed and converted into these electrons, electron hole pairs, which then undergo all these light reactions to so-called chemistry. And the process of this harvesting is also a funnel because one has different pigments going down and to the reaction center, the bottom of the funnel where the charge separation occurs. And these high efficiencies are displayed by systems as diverse as green plants, bacteria living in shallow pools, and also bacteria living thousands of meters. This actually one is 2 kilometers below the surface of the ocean. And of course there's no sunlight here, but there are hot vents, and they obtain radiation, basically thermal radiation, from the coming up from the interior of the Earth, and use it in the same way. So here's another remarkable thing about the green plants, and this is really mind-boggling. Photosystem 2, which is the light harvesting apparatus in a green plant, which is in about 50% of the green matter on Earth. This is, so the part that does the light, that absorbs the light from the sun are chlorophyll molecules, which make the plants green because they absorb in other regions of the spectrum. And if you calculate how much chlorophyll there is in a green tree, and this is something which one of my colleagues, graduate students, calculated from this beautiful picture by Picasso, you find out that in a typical large tree, there's only 500 milligrams of primary chlorophylls that are responsible for making these absorbing sunlight and making the electron hole pairs. So all this machinery that the plant has to enable it to harvest the sun's energy is done by a very, very small amount of material. So these are remarkable nanomachines, really, truly remarkable. So the big question was whether when the light is absorbed, whether the light would be converted into an excitation of this chlorophyll molecules represented here just by this green circle. And it used to be believed that this excitation would then just hop from one chlorophyll to another through the system until they get to the region where the excitation can be converted into an electron hole pair, and then a chemical reaction would occur. So this is a process which has this very high efficiency, known as a quantum yield. But now it's thought that this excitation doesn't really hop, it moves more like a wave. And let me show you what is really meant by this. So saying it moves like a wave is basically saying that there's coherence. And here's a very classical, very old-fashioned example of demonstrating what coherence is. There are these 5 gentlemen here on a pentacycle, and if they all pedal together in step, they will move forward. They will be pedaling coherently, and they'll actually have then constructive interference and go forward. However, so then they would be like this wave down here. If, however, they're like this, they're all pedaling incoherently, and one has its foot up and the other one has a foot down. That's incoherent, and they will not move forward. They will very rapidly fall off. So coherent addition of waves at different frequencies, so we'll give them beats. These gentlemen will fall off. And what we do in the lab is we do this multidimensional spectroscopy where we put in here 3 pulses of light, and we vary the central time between the second and the third. We create a spectrum, and the spectrum, the way in which the spectrum changes with the time here tells us what happens to the excitation. And this is a movie which I think will probably take a minute, but it will show you. It's a minute which represents something like 800 femtoseconds. That's 800 times 10 to the minus 15 seconds. And this illustrates very nicely what these quantum beats are. So this is one of those spectra shown now in 3 dimensions, like a mountain. There's a peak and a col, and so on. And now if we change that time between the second and the third peak, the form of this spectrum changes, and you see the peak starts to oscillate, and there are peaks coming out here on the side of the mountain, which are also going up and down and oscillating. Okay? And these are there's evidence for so-called quantum beating of the electronic excitation in this biological light harvesting system. So I'll stop that. They're also entangled, and this is one of these really completely non-intuitive features that Schrödinger gave a special name to, the entanglement. He called this the characteristic of quantum mechanics. And what is it, the entanglement? It means that there are non-local correlations between electronic states on one molecule and on another molecule. And as a way to visualize this, if you're not at all scientifically inclined and can't start anything with equations, is if you look at this so-called ambiguous cube at the top, the pair of ambiguous cubes, depending how you look at them, you'll perceive one of them as being in the forward direction or in the backward, oriented in the backward direction. And whichever one you first lock onto, you'll immediately perceive the other one as having the same orientation. So your mind, in a sense, is doing some kind of measurement of the ambiguous form of these 2 wire mesh cubes. You're projecting that ambiguous form onto this possible answer or this possible answer. And entanglement means that you would be doing this with 2 cubes that would be separated regardless how far apart, any possible distance apart. They could be, one of them could be on the moon, one of them could be on the Earth. Quantum mechanics tells us that however far apart they are, if they are prepared in this ambiguous state, a measurement on one will always cause the other one to have the same result. So this is something very, very non-intuitive. But it seems to be evident in photosynthesis. So those are things that we're now doing, or the community is now doing, with this understanding that we've gained from the quantum role. It's actually only a quantum role in a very small part of photosynthesis, but it might enable us to design artificial devices which, in the absence of competing biological constraints, would be very effective for conversion for solar energy. And now let me say a few words before I close about the magnetoreception. So magnetoreception refers to the ability of birds to fly thousands of miles across the Earth's surface, and they do so by detecting the inclination of the Earth's magnetic field. So the inclination changes, it's perpendicular to the Earth's surface at the north and south poles, and then it sort of varies very slowly, and it's basically almost tangential to the surface at the equator. And so here is a bird, which we know birds can measure this. The question is whether they measure this using what the physicists, quantum physicists' basic equation, that's the Schrödinger equation, so whether they're singing a different song than we normally think. And just as a reminder, birds aren't the only creatures that can detect magnetic fields. Actually, the American cockroach has also been shown to detect magnetic fields. So it's believed that so here's a hypothesis which uses quantum mechanics, whereby the absorption of photons by the bird's eye then activates a molecule called cryptochrome in the back of the rods cells of the eye, and inside that cryptochrome there is a system here which will separate. Again, there's an electronically excited molecule which then converts to a separated electron-hole pair, and in this case it actually makes 2 different electrons located on different molecules. And the dynamics of this, the correlated dynamics of the radical pair electrons on different molecules is one possible mechanism for getting the sensitivity to the magnetic field of the birds. So this is also, that's the details there, I won't show you that, but this is also an example of this entanglement, because we have here two electrons which are located on different molecules and are undergoing some kind of coherent quantum dynamics. So this is only a hypothesis. There's no real proof, because in this case it's very hard to make experiments. We certainly don't have any experiments on live birds. We have behavioral experiments on birds where we can show that they are sensitive to magnetic fields, but there are no biological probes of the live birds. There are probes of cryptochrome. It's been shown to be present in the bird's eyes, but the details of the mechanism certainly haven't been pulled out. So let me, we're out of time, let me just take those two examples. Let me maybe just show you this one beautiful creature, which is something which is a photosynthetic organism. It actually eats its food and then eats chloroplast from algae, and then it doesn't have to eat again because it uses the chloroplast in the algae to do photosynthesis. So that's maybe something for us to aspire to, and it also is something which makes us feel very humble, certainly as physicists. There's a quote from Albert Einstein: we really feel that physics is very primitive when you actually look at a living object. And so, if you feel humbled, then just behave like a child.